Good morning.
Lord.
Excellent. Get all this arranged. Let's go ahead and get started. Today is Thursday, June 22nd. We are down to 4 more lectures, as crazy as that seems. We have to finish off in lecture our protections of the central nervous system and finish off our central nervous system by talking about the spinal cord. But as we mentioned, we're going to pick up today with the brain anatomy where we left off, trying to work our way at least through the rest of the cephalons and identifying the structures and regions and the functions of those regions. We have a handful of assignments that are still due, and then again next week, 1 week from today, you have your lab and lecture exam, and then on Friday, you have your cumulative final exam. So again, that should keep us busy. All right, any questions on any of that before we get started? All right, excellent. And then let's pick back up where we left off. We had just finished talking about the telencephalon, basically the cerebrum, the largest and most variable region across species. Again, as you look at different brains from different animals, the biggest differences you see are in the cerebrum. As we've talked about before, we basically have the second largest cerebrum in relation to our body, with, of course, the dolphins being the gold medalists there, but they don't have thumbs, so we get to beat them up. But the rest of the brain, what we commonly refer to as the brain stem, is made up of the remaining cephalons. And these are much more similar across species because these tend to do much more basic life functions that you need to do, whether you're a person or a cat or a hippopotamus or anything else in between. Let's start first by talking about our diencephalon. So here is the diencephalon, and by far the largest structure of the diencephalon is the thalamus. The thalamus, like the hemispheres of the brain, are paired structures. There are these two big clusters that we can see here what it kind of looks like. It's a cluster of nuclei. So it isn't just one nuclei, it is many nuclei clustered together into a big structure known as the thalamus. You have two of them, one on each side, and they don't touch. There is actually a little bit of space in between. That space in between is yet again one of our ventricles, where we're going to have our cerebral spinal fluid. And one of the things that you should notice when you look at this this cluster of nuclei and their functions is basically the thalamus's function is to be a relay system for sensory information. Almost all of your sensory information coming from your body goes to the thalamus first to be processed before it's sent to the regions of the brain that they are processed by, or the regions of the cerebrum that they're processed by. I say almost all because there is one sense that doesn't go directly to the thalamus. And what was that sense that we've already talked about that doesn't go to the thalamus?
Smell?
Yeah, our sense of smell, absolutely. Remember, our sense of smell, as we talked about, goes straight to the limbic system first, which is why it's so strongly associated with memory and emotion. But everything else, touch, hearing, all of those, notice here on the back, there's this little green structure known as the lateral geniculate nucleus. That is where the visual input comes in. I mention that not because you need to know that. You don't need to know that for the exam, but the lateral geniculate nucleus is where I did the majority of my research to get my PhD, and my grant actually requires me to say lateral geniculate nucleus 3 times in class, and so now I've said it twice. So there you go. We're almost to the end of that. But this is that relay station, so all the information comes up from the spinal cord, comes up through the cranial nerves, comes to the thalamus, and then gets directed where it needs to go. So like I said, from the eyeballs, it would come into that lateral geniculate nucleus, and from there, it would go to area 17, that occipital lobe. And that's my 3rd time. Excellent. Here, we see that sagittal section. And again, we learned about the corpus callosum. We talked about that last time. We talked about how this is that septum pellucidum, that membrane that if we were to poke through it, we would be in that lateral ventricle. And that allows us to also see the space that is the third ventricle. Remember, as I mentioned, the two thalami do not touch. So this oval-shaped structure right here is the thalamus. And the thalamus do not, the thalami, plural, do not touch. And so there is a space in between, except that they do touch. There is one band of axons that actually connect the two hemispheres of the thalami together, allowing them to communicate to each other. So notice if you were to cut that and then look at it from the side, you would see the oval of the thalamus and then that little bit of axon sticking out. And notice, lo and behold, that's exactly what we see in this sagittal section. Here again is the thalamus helping to form the wall of our third ventricle. And then notice right here is a little bundle of axons that connect the two thalami together. And being the clever, sophisticated students that you are, who happen to have your handouts in front of you, what do you think we call that small bundle of axons that connects the two? There you go. Didn't even give me a chance to finish. Absolutely. That bundle of axons that connect the two hemispheres of the thalami together is what is known as the intermediate mass. So there is one small void we'll see in our third ventricle because there's this bundle of axons that do connect across it from the two thalami. But the majority of the thalami help to form the wall of the third ventricle, as do this relatively smooth surface right here. This relatively smooth surface, again, is the wall. And let's be careful. On the exam, I could have, this arrow's way too small. I could have an arrow pointing here, and I could, of course, ask you to identify the space. And as I just mentioned, this space is the third ventricle. But obviously, there is a structure that is making up the wall of the third ventricle. And this structure happens to be below the thalamus. And since it's below the thalamus, how do you think we identify it?
Hypothalamus.
Exactly. A hypothalamus. So arguably, we could do the same thing with an arrow like this. I could ask you to identify the space, and you would say third ventricle, because again, I'm representing this whole space, or the structure, and you would say thalamus, or I could do the same thing here. Now, this picture makes the hypothalamus look like it is a smooth, continuous structure. However, oh, this is why I like this. Notice here, if we look very closely, we can see in this space, this is the third ventricle. And notice they've done a really nice job of showing that little bit of that intermediate mass connecting this thalamus and this thalamus together. So we see that there. Excellent. All right. What looks like a smooth wall is yet again a big bundle of nuclei, clusters of cell bodies and dendrites that are located here below the thalamus, known as the hypothalamus. The hypothalamus plays a major role in regulating very basic life functions, coughing, hiccuping, heart rate, body temperature, things along those lines that are very important. However, that isn't the most important thing that it does. To understand the most important thing that it does, we have to look below the hypothalamus. Notice below the hypothalamus is a structure that is known as the hypophysis, or what is also known as the pituitary gland. And both of those are acceptable terms. Hypophysis or pituitary gland. And what is it that the pituitary gland does? Anyone know? pituitary gland is the master endocrine gland. It produces well over a dozen hormones. Many of the hormones are things like thyroid stimulating hormone. What do you think thyroid stimulating hormone does?
Stimulates the thyroid.
Stimulates the thyroid. See how easy that was? Excellent. Yeah, absolutely. So many of the hormones that are produced by the pituitary gland control or regulate the activity of our other endocrine glands. So it was known as the master endocrine gland. It controls pretty much all the other glands of the endocrine system. And for many, 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 many years, we thought this was the command center for the endocrine system. Until about 25 years ago. About 25 years ago, they realized that actually the hypothalamus controls the pituitary gland. So yes, the pituitary gland is the master endocrine gland. It is the president of the United States. It is the one that is in control, standing out in front, sitting there that everybody sees and everybody believes is in charge. But we all know that behind the scenes, the Illuminati controls the presidency. And that's basically what the hypothalamus is. The hypothalamus is the Illuminati controlling things from behind the curtains, right? He is the Wizard of Oz behind the curtain, controlling things that we didn't realize were going on. Excellent. And notice these two things are connected. The pituitary gland hangs like this jelly bean on the underside of the brain, and it is connected by a stalk-like structure, a bundle of axons that connects the pituitary gland to the hypothalamus. And what might that stalk of axons be known as?
Tracts.
I'm sorry, say again?
Tracts.
No, what do we call this bundle of okay, you're right. It is a tract, but this particular tract has a specific name. What is the name of this particular tract that connects the hypothalamus? Not the optic. Optic tract, remember, has to do with our vision. But notice if you didn't know it or you couldn't figure it out because it's it's on your list, it's the next item on your list, but it is the infundibulum. The infundibulum is what basically connects the pituitary gland to the hypothalamus. And that is one of the ways that the hypothalamus controls the pituitary gland. But let's talk about what Samuel mentioned, the optic tract. Oh, here we go. This is a nice view. Notice here, they have conveniently enough for us removed the jelly bean. The jelly bean would be that pituitary gland that would be sitting right here, coming off of the infundibulum, which they have shown us nicely right here. Notice, hold on, let's go back to, if you think back to ancient times, like when we were learning about the bones, notice here is the thalamus with its intermediate mass. Here is the hypothalamus. Here is the infundibulum, which connects to the pituitary gland or the hypophysis, which notice sits in the hypophyseal fossa, the sella turcica, which is a bone feature of what bone again? What bone has this U-shape sella turcica in it that the pituitary gland sits in? Come on. I'll wait if you have to look it up. There you go, sphenoid, perfect, excellent. Absolutely, excellent, excellent, excellent. All right, so orienting ourselves, this is an inferior view of our brainstem. As I mentioned, they have removed the pituitary gland, so we don't have to worry about looking at that, so we can see the infundibulum. Notice also here we see the two mammillary bodies, and now you can see why they got their name, because they look like tiny little boobies on the underside of the brain stem. And remember, those, as we talked about, are what provide us with that smell and taste aversion, connecting via the fornix to the hypothalamus. And that allows us to see 3 additional structures we are responsible for as well. Starting first with the 2 optic nerves. Optic nerves, as we've talked about, are those bundle of axons that carry information from the eyeballs towards our brain. And remember, as we talked about, that is, or we haven't talked about, but we're going to talk about, that is, I'm not sure why that is N2 there. Cranial nerve 2, that optic nerve. But remember, as we talked about, we have this issue with sensory information, where all of our sensory information has to go contralateral. So things I feel from the right side, I process on the left. Things I hear from the right side, I process on the left. And things I see, not from the right eyeball, but from the right side of the visual world, need to be processed on the left side of the brain. So remember, as we talked about, we have this region where there is a partial crossing of the axons. And this region with the partial crossing of the axons is what is known as the optic chiasm, which I don't know if you can see. I don't know if you get to see my controls, but they're in my way. So if I move those like that, then we can see that optic chiasm where they cross. Remember, as we talked about these cranial nerves, the optic nerve is eye-specific information. What do I mean by that? Well, again, as we've often talked about, one of the easy ways to understand how these systems work are to damage them. If you were to cut one of your optic nerves, that would basically be the same as putting your hand over one of your eyes. You would receive no light information from one of your eyeballs. If, however, you cut these bundles down here are what are known as the optic tracts. And these optic tracts contain side-specific information. If you were to cut the left optic tract, basically you would see nothing from the right side of the visual world. It wouldn't matter which eye you used. If you used your right eye or your left eye, it would be the same. You would see nothing on the right side of the world. We have, this again does a really nice job of showing the brain stem. And we can see those structures here, but here we get a better view of this visual pathway. Notice again, as we see, we have eye-specific information coming out of the optic nerve. We get a partial crossing of information where some axons cross and some axons do not. And then that brings us to our side-specific information, which is the optic tracts, which carry the information to the thalamus to be processed, because remember that thalamus is the relay station. And lo and behold, your textbook here shows that it's the lateral geniculate nucleus. And hey, look, we got it in an extra time. And then from there, the information goes to the optic nerve, to the occipital lobe to be processed. And I think I have one more. That original picture we showed, it shows how everything from the left goes to the right and everything from the right goes to the left because of that partial crossing that takes place in the optic chiasm. One more thing. Excellent. All right. One more thought. Remember we talked about the deficiencies that would occur if you damage this. If you were to cut an optic nerve, that'd be the same thing as basically just ripping an eyeball out of your head, or less extremely covering an eye. If we cut that optic tract, then you lose side-specific information. Well, what happens if you cut the optic chiasm? It turns out if you cut the optic chiasm, what ends up happening is you get a tunneling of your vision, where you lose all the peripheral information. Now, clearly, this is in a place inside of your brain where you can't cut the optic chiasm without having severe damage to other parts of your brain. However, let's look at the orientation of this. Notice this structure right here is that optic chiasm. That is that bundle of axons where we have that partial crossing taking place. And right beneath it is the pituitary gland. While people typically don't tear their optic chiasm, there are some conditions that can cause your pituitary gland to swell. And remember, your pituitary gland is sitting within that bony U-shaped sella turcica. So what happens when that pituitary gland enlarges, it actually can push up and squeeze the optic chiasm. So there you are sitting on your couch, and suddenly you get this tunneling of your vision. You go rushing to your optometrist to try to figure out what's wrong with your vision. And can your optometrist do anything to help you?
No.
No. I mean, they're horrible people to begin with. Don't even get me started on optometrists. We'll talk about them later. No, obviously you need to see an endocrinologist having to deal with your pituitary gland, having to deal with your hormones. But it expresses itself with this very distinct visual deficiency. All right. Questions on that? All right, there are only a couple more structures of the diencephalon we need to identify. Remember earlier, last class, we talked about how this big C-shaped, comma-shaped structure is that corpus callosum that we said was the largest of our commissural tracts, bundles of axons that go from one hemisphere to another. And of course, the key word in that sentence was the largest, because that implies that there are other commissural tracts. And in fact, there are 3 that you are responsible for. We've already talked about one, the intermediate mass, connects the 2 hemispheres of the thalamus. But there are 2 others that you need to know as well. This one right here is what is known as the anterior commissure. It is a commissure because it is a bundle of axons between the 2 hemispheres of the brain, and it's the one in the front. Notice it's above the optic chiasm. However, the easiest way to find it is to find the fornix. The fornix always points right at the anterior commissure. We'll see it here. We'll look at the chart and see it there in just a moment. And anytime you have an anterior something, you of course are going to have a posterior something. And lo and behold, directly behind the anterior commissure is the posterior commissure. Again, these things don't have functions. They're just structures, bundles of axons that connect the 2 hemispheres together. Notice the other way to find these is to use the corpus callosum. Notice the corpus callosum comes around and leads towards the anterior commissure. It comes around and leads towards the posterior commissure. However, if you also notice, to get to the posterior commissure, there's something else in the way. Any idea what this something else in the way is? It is sometimes referred to as your third eye. It is also referred to as your first chakra. Anyone have any idea what this particular structure happens to be? Again, we're running out of...
Pineal body.
Pineal body.
Yeah, this is the pineal body, absolutely. What does the pineal body do for you?
Pineals.
That's a good guess compared to the names of some of the other things. No, nope, but I figured somebody would know. I know you know, even if you don't know, you know. Somebody here has got to be aware. The pineal body is also known as the pineal gland because it produces something. What is the hormone that is produced by the pineal gland?
Is it melatonin?
Yeah, Arabella, and I didn't hear who said it. I also got it. Melatonin, excellent, right? That's the thing that makes your skin dark, correct?
No.
No, what is melatonin then?
Helps you fall asleep.
It helps you sleep.
Yeah, it plays an important role in your sleep-wake cycle, right? Notice how easily we take that information for granted. We didn't always know that, right? There was a mystical, magical time, ancient time known as the 70s. And in 70s, when young people like yourselves graduated from high school, they all went to four-year universities or the Army. And when they went to a four-year university, their first year at that four-year university, they were required to take a psychology class. And that psychology class was pretty much just an excuse for the professors at the university to manipulate and torture these students. We've all heard of many famous cases. The prisoner's dilemma from Stanford is a classic example of how this went wrong. But basically, it was this mystical, magical time where we could do pretty much anything that instructors wanted to do to freshmen. And one of the things they wanted to do was figure out if we could tell time without watches. All right. How many of you got up this morning because you wanted to get up? Yeah, see, nobody spoke up, right? Your alarm went off, or your kids or your dog woke you up, or the garbage truck woke you up, or you got up because you had this class, right? Most of our scheduling are driven by external cues, right? When we eat, when we get up, when we go to sleep, those things are all driven by basically outside sources of stress that we have to deal with, whether it's work or jobs or kids or families or whatever. However, right, birds don't have watches. You know, wolverines don't have calendars, and yet they seem to know when to get up, when to eat, when to migrate south for the winter. So what they were curious in doing is they took these first-year freshmen, and they took a bunch of them, and they locked them each into their own individual room in a basement. They had no windows. They had no clocks. They had no televisions. They had no way of being able to discern time. They basically had a bed, a refrigerator, and a light switch that they could control. And they were curious what would happen. And as it turned out, after a couple days, people get on a fairly regular cycle. Removed from all those external stimuli, a person eventually will reach a state where they typically get up at the same time every morning, they eat at the same time every day, they go to sleep at the same time every day. In fact, what they found is most people actually were on a 25-day cycle, not a 24-hour, 25-hour cycle, not a 24 like we have time-wise, but we all have this cycle. And it turns out it's this hormone, as you guys mentioned, melatonin, that helps us to regulate that cycle. Now, hopefully none of you have been locked into a basement for a week and a half, but many of you have experienced the disruption of this because, again, we just got a weekend coming up. So on that weekend, even though it's the weekend before your exam, you may want to fly to Paris for the weekend. And when you get to Paris, you're getting off the plane and you're ready to have dinner and party. And everybody else is waking up and getting ready to go to work, right? What do we call that disparity of sensations?
Jet lag.
Jet lag.
Jet lag, absolutely, jet lag. And that is because of this disruption or this non-synchronous aspect to your melatonin and the melatonin of the people that are there. Now, remember, I also called this pineal gland your first chakra and your third eye. because as they found, there are actually direct connections from your retina to the pineal gland. And again, the more we understand the brain, the better we can use it to our advantage. If you are traveling to France or someplace else for the weekend, one of the best things you can do is to get up at sunrise, sit outside, and take about an hour of sunlight in. As you sit out there and take about an hour of sunlight in, that sunlight from those visual connections to your pineal gland help to reset your melatonin levels and can dramatically decrease the symptoms of jet lag, because you are reestablishing an appropriate hormone level for that new environment that you are in. Excellent. So that is the pineal gland. And notice, as we mentioned, from the corpus callosum, that's how we got to this in the first place. From the corpus callosum, we pass the pineal gland, and then that brings us to our posterior commissure. And I believe that is everything on our list for the diencephalon. Is that correct?
Yeah.
Excellent. Here we've got some more pretty pictures. Notice here we very nicely see our corpus callosum. Notice here is our fornix, and remember, as we mentioned, our fornix points straight to the anterior commissure. Here we see that membrane. As I mentioned, if we poked through it, we'd be in the lateral ventricle. That is our septum pellucidum. And then again, from the corpus callosum, here is our pineal gland. And then this right here is your posterior commissure, right across from the anterior. Excellent. Last, again, view from the real brain. Notice we can also see those optic tracts and the optic bulbs, olfactory tract and olfactory bulb, optic nerves, infundibulum. Again, our pituitary gland is missing. Little mammillary bodies, optic chiasm, and the optic tract. So these are all things that can be seen from these different views on a real intact brain as well. All right, we did that. We're here now. All right, excellent. I believe that is it for the diencephalon. Questions on that? All right, mesencephalon. The mesencephalon is basically, notice we make a cut here below the mammillary body, below the posterior commissure, and above the pons and above the cerebellum. And this narrow region in here, the smallest region, is the mesencephalon. And again, mesencephalon is still a perfectly acceptable way to identify it, but it is also sometimes called the midbrain. One of the easiest way to see the most distinct structure is indeed in this view. Notice there's this big, huge structure that is white in color on our illustration, and of course that indicates axons. But notice if we actually take an anterior view of this, here in green, these big bundles of axons heading up from the spinal cord up towards the cerebrum, this major pathway of axons going up to the cerebrum is what is known as the cerebral peduncle. You see it very nicely in this anterior view. However, one of the best ways to actually see this is probably in a cross section. Notice in a cross section here, we very clearly see these two big enlargements, and we see that they are very white in color. And these two big enlargements are, again, as we mentioned, what we call the cerebral peduncles. And their job is basically to carry information to and from the cerebrum. One of the things to remember about the brainstem is it has the ability to process information, make sense of information, but it still has to carry information to and from the cerebrum. And we'll see the same thing in the spinal cord as well. It has gray matter where it processes information and makes decisions, but it also has a lot of white matter carrying information up and down to the brain. These cerebral peduncles are also useful because it helps us to identify the second region we need to be able to identify here, and I will use yellow to highlight this. Notice there is a very dark region directly posterior to the cerebral peduncle. This very, very dark region is known as the substantia nigra, basically meaning black substance, because it was a very dark, very distinct, very easily recognizable region of the brainstem. Except in people with Parkinson's. People with Parkinson's disease don't have a substantia nigra, or they have it, it's just not dark in color because they're lacking a very important substance which is found there. And what was that substance we said people with Parkinson's didn't have? Caused them to have that shaking. Then we discovered the L-dopa pill that would allow them to get it and make their own. What was that neurotransmitter?
Dopamine.
There we go. Dopamine, exactly. Arabella and Jacob got it. Absolutely, dopamine. Yeah, the substantia nigra is just filthy with dopamine. Massive, massive amounts of dopamine in there, and it gives it an incredibly dark pigmented color. And again, that's one of the primary ways we actually diagnose Parkinson's. Obviously, you can't diagnose Parkinson's until someone is dead because most living people don't want you to cut their midbrain. But when a Parkinson's patient dies, one of the things they do is look at the substantia nigra, see that it is clear to confirm that indeed they did have Parkinson's disease. Now notice the rest of this looks a little bit more uniform. It can be a little bit more challenging to differentiate the regions, which again is one of the nice reasons why we're able to use with a PowerPoint presentation, or in this case, an online presentation of the lab exam, illustrations. We don't just have to look at the cut nervous tissue. Notice again, for whatever reason, they've got this one turned upside down. So again, anterior, ventral is down this way. And we have our big, huge knuckles that are the cerebral peduncles. We have our nice, dark, distinct region that is the substantia nigra. Notice deep to that is a structure called the red nucleus, which you are not responsible for. But deep to that, you will notice there's this shaded area right here. And this shaded area right here is a region that is known as the reticular formation. The reticular formation is a very interesting region of the brainstem that plays a very important role in our alertness, our awareness. I'm not talking about being tired or being restful, right? That's melatonin, that's the pineal gland. We just finished talking about that. What I'm talking about here with the reticular formation is that my beautiful wife can lay in bed next to me while dogs are barking outside and sirens are going off, right? Shotguns are going off in the background, and she sleeps soundly, snoring away blissfully without a care in the world. But, all right, I step out of bed and there's that little creak of the floorboard as I'm on my way to go play Xbox, and she's instantly awake. Or for all you that have had newborns before, like I said, complete chaos can be going outside of the house and she sleeps blissfully through it. But that baby whimpers just a teeny bit, and what happens to mom?
She wakes up.
Instantly awake and aware. Notice the difference between those. It isn't the amplitude of the sound or the quality sound or anything like that. It is the importance of the sound. That wimping baby, especially with your first kid. Second kid, you couldn't care less about. But with the first kid, right, just the tiniest little whimper is vitally important. Important piece of information. You are instantly alert and awake. Right? That dog barking outside doesn't mean anything to you. So even though it's loud, it doesn't cause that same alertness, and you're able to sleep through it without any problems at all. right? You see this a lot with siblings. Again, that with the often a baby and an older sibling are put in the same room together. So what happens is the baby wakes up screaming and yelling during the night, waking up and disturbing mom and dad, but the sibling sleeps right on through it. Because again, it isn't the amplitude of the sound, it's the importance of it. And so this reticular formation is the area that is involved in that awareness, that alertness. The flip side is true as well. Activation of your reticular formation can also affect your ability to sleep. So for instance, this is another area that can be sensitive to light. If it is too bright in your room, or if you have the TV on, or you're staring at the white light from your phone, that can be stimulating your reticular formation and causing problems, difficulty falling asleep. Right? Again, the more we understand this, the more we change things. Back in ancient times, and by ancient times, I mean when I was a kid, if you were a kid, most kids are some wariness of the dark, so you get them a night light. And in my days, a night light was this little light bulb you plugged into an outlet, you flipped it on at night, you flipped it off first thing in the morning, right? Or you just left it on all the time because during the day it didn't matter. Right? Well, what they found is if there's too much ambient light in the kids' room, right, that can stop them from getting deep, restful sleep. So nowadays, most lights are things, well, like pillows and stuff like that light up and shine stars on the ceiling or things along those lines. But most lights these days have a timer where it'll go on for 20 minutes, long enough for the kid to calm down and fall asleep. But then after the 20 minutes, it turns off so that the room is darker to help to encourage more deep, restful sleep. Because that light can stimulate the reticular formation. All right. So don't watch TV, don't play on your phone when you're trying to go to sleep. Questions on that? Excellent. Two more things, really three. Notice, and let's go back to this picture for a second. We know, we can't see it, but we know the lateral ventricle is to the side there. We have a third ventricle right here. And even though we haven't talked about it yet, here between the pons and the cerebellum is a fourth ventricle. Well, cerebral spinal fluid has to be able to get from the third ventricle to the fourth ventricle. So notice there is a little fluid-filled tunnel that passes through the midbrain. I mention that because if we go back to this cross-section picture, here we see it really, really nicely. Notice there is a hole in the center of our mesencephalon, our midbrain, where that fluid goes from the third ventricle, this space here between the thalami, to the fourth ventricle, this space here between the pons and the cerebellum. This is known as the cerebral aqueduct, or the aqueduct of the midbrain. Aqueduct, of course, is a passageway for fluid, right? Southern California uses aqueducts to steal water from us northerners, right? So we have that there, and we see that example of it here. And we also see that, again, aqueduct of the midbrain or cerebral aqueduct, both of those are acceptable terms. The last structures we are responsible for are these bumps that we see on the back. Notice they're not as prominent as the cerebral peduncles here. And yes, it kind of looks like two when we look at it from a cross section. But where you see it best is when we look at it from a posterior view. Notice in this posterior view, we see that there's not two bumps, but actually four bumps. These four bumps collectively are known as the Coppora quadramina. All right, quad, four, so the Coppora quadramina. These, notice as we look here, are nuclei. That play an important role in coordinating information. Again, I have to work on my analogies. Normally at this moment in the class, I've already talked to a student who's sitting in the back of the class, and that student in the back of the class takes their textbook and slams it on the ground right at this period of time. I was going to call all of your significant others and ask them to do that at this moment during the middle of lecture, but I wasn't able to get a hold of everybody, so I just figured it wasn't worth it. But all kidding aside, if someone were to drop a book and make a loud noise in the back of the classroom, what would we all do?
Turn and look.
Yeah, we would all turn and orient to it. And it's a process that we take tremendously for granted. Heck, picking up that glass of water is something that I take for granted because I see that glass of water and I'm able to reach out and grab it and bring it to my lips. Now, I'm able to do that because I'm able to observe the glass of water. Light bounces off that glass of water into my eyeballs and hits my retina. And we think about where something hits our retina and our eyeball tells us where it is in space. But here's the problem. My eyes move. I can see the camera and the light from the camera is hitting my eyes right now, but I have the ability to turn my eyes. And as I turn my eyes, the light from the camera is hitting a different part of my eyes. And I can move my head. As I move my head and I move my eyes, that light from the camera reaches a different part of my eye. So for me to know where that camera or where that glass of water is in space, I need to be able to do two things. I need to know where light is hitting my retina, but I also need to know where my eyeball and where my head is located in space. And body, because I can turn my body as well. So notice what I need to be able to do is coordinate vision and where my body is in space. Body, head, eyes are in space. And that is a tricky process. But I could do the same thing when my phone rings. When my phone rings, I hear the phone and I'm able to reach out and grab that phone. But again, where that sound is coming from depends on where my head or where my body is located. I'm not a cat. I can't move my ears the same way I move my eyes, but I still have to be able to coordinate auditory system, hearing, and where my body is in space. Body and head. Again, I can't really move my ears, so that's not as much of an issue. So I have this process where I need to coordinate this information and put it together. And that's what this Coppora quadramina do. These four structures collectively coordinate sensory information. Specifically, vision and where I am in space is the job of the top two nuclei. And those top two nuclei are known as the superior colliculi. Colliculus is the singular, but you have two of them, so they are the superior colliculi. And coordinating hearing and where my body and head in space is the job of the bottom two nuclei. And so not surprisingly, those are called the inferior colliculi. So collectively, these four things are called the Coppora quadramina. But really, they're made up of four nuclei with two different jobs. The two top ones coordinate vision and where our body is in space. The two bottom ones coordinate hearing and where our body is in space. So collectively, the Coppora quadramina are made up of two superior colliculi and two inferior colliculi. Questions on that? All right, excellent. From there, we are done with we've done now our telencephalon, diencephalon, and mesencephalon. We have two more cephalons to go, but like this rest of this week, we have a lot of material to cover and a lot of time it's going to take to do it. So again, I think shorter periods of discussion and more breaks helps to keep the information fresh. So let's go ahead and take our first break. We will restart at 9:19. That'll work. And I will start the recording at that time. All right, excellent. So 15-minute break, and then we'll come back. Questions on that? All right, hopefully get some coffee, get some caffeine, get something, and then be ready to roll when we get back. All righty. Let us move down to the mesencephalon. Again, the way I always remember it is met has a T in it. And this is the portion of the brainstem that has the two big enlargements. Here we see the first big enlargement. It is the pons. So the pons, again, like most of the brainstem structures, are comprised of a bunch of bundles of axons going from one direction to another, and a bunch of clusters of nuclei. Two of the most prominent nuclei, as you can see right here, are ones that you don't have to know by name, the pneumotaxic and the apnoistic centers. And if those names mean anything to you, then you're aware of the fact that they play a huge role in determining your normal resting breathing rate. So not me who's sitting here talking, but you who is sitting there quietly listening and breathing, hopefully while you're doing that, that normal resting tidal breath that you're taking is primarily being controlled by these two major regions found in the pons, with a little help from your medulla oblongata as well. So there's some of that going on. And like I said, there's bundle of axons. Yes, there are axons that travel again to and from your cerebrum up and down the brainstem. But if you notice over here to the side, there is not one, not two, but three large bundles of axons that have been cut that actually connect from here on the pons to the cerebellum. And we can see a little bit of it up here. Notice up here we see the cerebral peduncles. That is going to and from the cerebrum. Well, these three bundles of axons happen to be going to and from the cerebellum. So guess what they are known collectively as?
The middle cerebellar peduncle.
Well, you are correct, absolutely. They are the cerebellar peduncles collectively. But you are correct. And even though this illustration, this one doesn't show it as nicely, notice the one over here to the side, and the other view we'll see in just a minute shows it nicely. Not all of the cerebellar peduncles are equal. The biggest, the largest, and therefore the one that is most important to us is the middle cerebellar peduncle. So the others, and here I think I've got another view of it here. Here we go. Notice there is a superior, there is an inferior, but it is the middle cerebellar peduncle which is the largest, and the easiest to see and identify, and the one you will be responsible for on the exam. And I think, hold on, I'll have to cheat for a second. See if I have, I don't think I have that picture here. Oh, you know where they might have it? Hold on. Let's see how good that CRC site is. Here we go. This will work. So on that CRC site, under the brain, here we see the brain stem or portions of the brain stem. Again, this is one of the models that is in the classroom. Notice we can really nicely see that pituitary gland and the infundibulum hanging down. Here we see a little bit of the thalami, and the insula is actually located over here as well. Here is the pons that we're seeing from the front. And notice if we turn it around to look at the back, notice the cerebellum has been removed. But these two big white surfaces located here on the side are those big bundles of axons carrying information to the brain. Let's see if they have, nope, they don't. Oh, but that's good to know. So here is the pons from the front, and this posterior view does the best job of showing it. This big white mass to the side is that largest of the cerebellar peduncles, the middle cerebellar peduncle, which we can see in a couple of locations. And again, bundle of axons, so nothing too particularly special that we need to know. That leads then to our other big enlargement, which is the cerebellum. The cerebellum we've also looked at a little bit before when we talked about it when we were doing the histology. Remember, we talked about how the gray matter is superficial, the white matter is deep. The white matter forms this elaborate tree-like branching structure. And so what is the white matter of the cerebellum collectively known as?
Arbor vitae.
The arbor vitae, excellent. So we see that nice arbor vitae there. We see that it also has ridges and grooves. The grooves, just like in the cerebrum, are known as sulci. However, remember here, the ridges are not gyri, they are folia, because they thought this looked like the edges of a leaf, the ruffles of a leaf, so like foliage. All right, so that's anatomy we've already learned. What we didn't talk too much about is some of the gross anatomy. Here in an intact cerebellum, here we see an actual cerebellum, and here we see an illustration of it. It always reminds me of like a pound of ground beef that you get when you go to the market. We can see those folia, we can see those sulci, and we can see, like the cerebrum, there are two hemispheres. Two hemispheres to the cerebrum, two hemispheres to the cerebellum. The big difference between the two is how they connect. Notice, as we've talked about, there is a big, huge, deep invagination, that longitudinal fissure that almost completely separates the two cerebral hemispheres from each other. But notice the opposite is true on the cerebellum. On the cerebellum, there is actually a raised ridge that connects them together. Here we see that raised ridge. So there's no deep invagination, instead there's a raised ridge. And here we see the nice simple illustration of it as well. Notice this raised ridge, and I have to cheat a little bit and pull that out so it's out of the way, is made up of a bunch of folia and their gyri in between, and pardon me, and their sulci in between them. And so basically it looks like a long, thin, thread-like segmented structure. It essentially looks like a worm. And that is actually where it gets its name. It gets its name from the same place that the vermicelli noodle gets its name, because it is a worm-like noodle. Well, the vermis is a worm-like structure that holds the two hemispheres of the cerebrum together. So there's our vermis. Our vermis is this worm-like segmented enlargement that holds the two hemispheres of the cerebellum together. All right, I think that is all of the anatomy we need to know. So we've talked a lot about the anatomy of the cerebellum. What we haven't talked about is the function of the cerebellum. The cerebellum plays a huge role in helping to maintain, let's say this way, how do I want to say this? I don't want to say maintain, because it's not like our autopilot. Basically, its job is to fine-tune learned muscle behaviors. So for instance, this includes things like posture. This includes things like walking, right? These are types of fine muscle behaviors, learned muscle behaviors, and it allows us to fine tune them. As we've talked about, or refine these movements. Again, one of the things that we've talked about in the past is one of the easiest ways to understand how these areas work is to see what happens when these areas are damaged. It is not uncommon for an individual, when they're in a car accident, if they do not have their headset or their headrest set properly, where that headrest, as their head is slammed back into that headrest can hit them right in the back of the head where that cerebellum is located. When that happens, it disrupts the activity of the cerebellum and leads to a condition we call ataxia. Ataxia is where we can't refine those learned behaviors. So let's think about this. You've just been in a car accident, you've taken a blow to the back of your head, and now you're feeling somewhat dizzy. A policeman shows up and asks you to get out of the car and stand up. And can you stand up straight and maintain your posture while he's talking to you?
No.
No.
No. And then he asks you to walk a straight line. And are you able to walk a straight line?
No.
No. There have been plenty of instances where people have been arrested on suspicion of drunk driving, and then they get to the police station and they blow in the breathalyzer and they blow a 0.0. And the reason for that is they're not drunk, they've had a traumatic brain injury. They've had this blow to the cerebellum that has disrupted their ability to stand upright and walk properly and maintain their balance. The good news, in most instances, it is temporary, and people frequently recover shortly from ataxia. But in some rare instances, the trauma can be permanent. Imagine how horrible that would be. All the downsides of being drunk without any of the fun parts of it. Right? You can't stand upright, you can't drive, you can't walk in a straight line, but you can still think perfectly fine, right? I shudder to imagine what that would be like. So again, that's a perfect example of, again, it helps us to understand what this area does. All right. Questions on that? Excellent. With that, then, we are down to our bottommost area. Oh, and again, while we're here, and we'll emphasize this again a little bit later, but again, notice here between the pons and the cerebellum is also where our fourth ventricle, that fourth fluid-filled space, is located inside of our nervous tissue. All right, excellent. That brings us down to our Mesencephalon. The same way that the terms telencephalon and cerebrum are interchangeable, the terms Mesencephalon and medulla oblongata are interchangeable terms as well. The medulla oblongata, not surprisingly, is the base of the brain. Right? Base of the brain stem. And so not surprisingly, it is responsible for many of our very basic life functions. Things like breathing, like we talked about, cardiovascular control, controlling our heart rate, vomiting, swallowing, sneezing, many of these very basic like functions that you and your dog and a panda are all capable of doing occur here in our medulla. We could spend the rest of the semester talking about just what's going on in the medulla. It is a very, very important structure. And as you can see from the illustration, there are both a large number of axons traveling through it, but also a very large number of nuclei. However, we'll just focus on two to make this a little bit easier. Notice when we look at this anterior view of the brainstem, there are these two big, very prominent enlargements that stick out in the front. These two prominent enlargements are what are known as the pyramids. The pyramids are major bundles of axons. But there's something really important about these pyramids. Here in the pyramids is where the majority of our sensory information crosses. Remember, as we've talked about, if I touch something with my right hand, I process that with the left side of my brain. If I hear something with my right ear, I process that with the left side of my brain. Here in the pyramids, this is the primary location where the majority of that sensory information crosses. So here in the pyramids, this is actually where information goes from one side to the other in our brain. So that's a useful and important thing to notice. Notice then, and again, we're not there yet, but we will get to it. On both sides of the pyramids are actually these bundles of axons, and these bundles of axons actually collectively are our cranial nerve 12. We'll talk about cranial nerve 12 a little bit later when we get to the cranial nerves, but I'm pointing it out to you now so that we can orient ourselves, because that's cranial nerve 12. Because notice over here to the lateral side of the brainstem are three more cranial nerves. These three cranial nerves are cranial nerves 9, 10, and 11. Again, we'll talk about them. We'll talk about what they do. We'll do all of that in a little bit. But the reason I point out cranial nerve 12, the reason I point out cranial nerves 9, 10, and 11 is because if you look closely in between them, what would be a good color from here? Blue. There is an enlargement right here in between them. So notice this big enlargement right here to the sides of the pyramids in between all these cranial nerves. And this enlargement is known as the olive. Notice the olive is the largest nucleus of the medulla oblongata. And the olive is responsible for proprioception. What the heck is that? Anyone know what proprioception is? No one? Or no one is willing to speak up? All right, well.
Awareness of our body position.
Exactly, absolutely, right? Assuming I haven't had too much vodka already this morning, with my eyes closed and my hands all the way out, I can touch my nose without my eyes being open. I'm able to do that because I have an understanding of where I am in space. If I have my eyes closed, maybe I've had a little too much vodka. If I have my eyes closed and I'm standing versus laying down, even with my eyes closed, I can tell that. Because I know where my body is in space. That sense of proprioception is understanding where our body is in space, right? Much like everything else, it is a learned behavior, knowing where our body is in space. This becomes significant when we're a baby, learning how to stand, learning how to walk, getting an understanding of where our body is in space. But it's also a big deal in puberty, because there you've got that 14-year-old boy walking down his hallway to his bedroom, the same way he's done a million other times. And right there on the end table is, grandma's vase, the one that's been in the family for generations. And he goes walking down the hall, he hits it and knocks it to the floor. Does he do it because he's an asshole? Well, possibly, right? Or he could just be walking down the hallway and fall because he's on drugs. Well, maybe. Or more likely, it's the fact that in the past year, he's grown 3 inches. And as he's grown 3 inches because he hit puberty and his body is starting to change, he has less of an awareness of where his body is in space. So often during puberty, especially in boys, because they tend to grow more rapidly than girls, they become less coordinated for a brief period of time, where they can just be walking down the hall and trip and fall. Or because their arms are now longer than they were before, as they're swinging their arms like they normally do, they knock something off the table because they're not aware of it, or they trip over something because they're not aware of it, because that changes the body, and it changes our understanding of where the body is in space. Again, doesn't mean he's not on drugs, doesn't mean he's not a jerk. Those things are still possible, but it could just simply be that he has less of a sense of an awareness of where his body is in space. And that's what our olive does. Our olive tells us where our body is in space. Notice this olive is one of the things that communicates to those superior and inferior colliculi that we talked about, understanding where our body is in space and coordinating it with vision and coordinating it with auditory. And which colliculi were the ones that did vision? Superior or inferior?
Here.
Superior, excellent. And so
Superior.
sound is inferior. Excellent.
Could there be a correlation between somebody's olivary nucleus and them being like klutz?
Can be, absolutely. So yeah, less coordination with where their body, less communication between the two sides, absolutely. Or again, like I said, puberty can have a huge effect. I think I've mentioned it in this class before. My brother-in-law, his senior year in high school, grew 12 inches. Not that he was super coordinated before, but he was really less coordinated for a time after that. That's some very rapid growth, and it's very hard for while our brains are plastic at that age, it's very hard to reorganize the body that rapidly to have an understanding of those rapid changes. So not surprisingly, he was fairly klutzy during those times. So yeah, I could see that. All right. Not that I knew him then, but I've seen videos. All right. Questions on that? Well, there you go. Just that easily, we have identified all of the structures of the brain and brain stem that we are responsible for on this exam. All right, questions on that? All right. The last thing we need to do to finish off the brain is to kind of summarize some of this information we have already been talking about. And that is the protections of the brain. We actually mentioned these on the very first day of class, and so we've come full circle and are talking about them again. And if you remember, on the first day of class, we were talking about how the brain and the spinal cord, or at the time, more specifically, we were talking about the organs in the dorsal body cavity, have a special collection of connective tissues that provide protection for them. And collectively, those layers of connective tissue are known as the meninges. Remember, we went into then talk about how in the ventral body cavity, it is the serous membranes that protect the lungs and the liver and the heart and things like that. But the brain and the spinal cord are protected by 3 layers of connective tissues known collectively as the meninges. And then the meninges, like phalanges, is the plural. And so, of course, like phalanx, the singular is menix, which I thought I had here. Where do I have that? I guess do I not have that? Thought I had that. Oh, it must be on the other one. So here, let's do it this way. There you go. So again, 3 layers of protective tissues. And those 3 layers of protective tissues collectively are known as the meninges, the singular of which is menix. The first of these is what is known as the dura mater, the tough mother. Notice, let's look here a little bit at the anatomy. Clearly, the scalp is representative of mine with just a little bit of hair on top of it. But more importantly, here is something we understand. Here are the skull bones forming the cranial cavity. We know that this is a flat bone with a cortex on the outer surface, the diploe, the spongy bones on the middle, and most important of all, a layer of dense irregular connective tissue that surrounds the bone, acting as a attachment point for blood vessels, providing some protection. And again, there's really no tendons connecting to this here. But that dense irregular connective tissue we called what based on its location?
Periosteum.
Excellent. That periosteum. Notice the outer periosteum is the outer periosteum, but notice the dura mater is actually comprised of 2 layers of thick, dense, irregular connective tissue, and the outermost is the internal endosteum. By having these big 2 thick layers, thick layer 1, thick layer 2, it provides a tremendously thick, hardy layer on the outer surface. If you want to do brain surgery on someone, you have to cut this leathery structure with some scissors or a scalpel, and it is robust enough where after your surgery, you can actually sew it back together, closing it up. And notice the advantage of having one of those 2 layers being the periosteum is it actually holds this protective bag to and attaches it to the skull, helping to stabilize it and hold it in place, helping to anchor the brain there. So we have those 2 big thick layers. But notice the advantage of having 2 layers. There are parts of the brain where the innermost layer of the dura can actually extend off. This does 2 things. It allows it to form these layers of dura mater that can penetrate deep. Like in this case, in the longitudinal fissure, that can provide extra stability to the brain, helping to hold it in place. So it's not just a plastic bag around the outer surface. It actually has some invaginations that stabilize it in place. And the second big advantage of this we see right here. In between the two layers, we can form a big space, and that big space is known as the dural sinus. Now, notice when we talked about sinuses before, we were talking about our bones and they were air-filled sinuses. This is actually a blood-filled sinus. A sinus is the name for a very large, a very loose vein. And I'm not sure we've done this in this class, but how do you define a vein?
Carries oxygen, rich blood, or the opposite.
So you are correct in that most veins do indeed carry oxygen-poor blood. However, the key word in that sentence was most. And if the answer is most, that means not all. And so if it's not all, is that necessarily the best definition we could have? It's useful because it's right most of the time, but most doesn't help us all the time. We need something that can help us all the time. Is there something that all veins do?
Transport
Carry
blood.
blood.
Yeah, and it's where they transport blood. All veins carry blood. Which way? To the heart or away from the heart?
To the heart.
Excellent. By having these big, loose sinuses in our cranial cavity, it really helps us to drain blood, drain fluid from the cranial cavity. Why is that so important to get that blood, to get that fluid out of the cranial cavity?
Because you need to bring in fresh blood.
True,
Oxygen.
absolutely. We need to bring in fresh blood. But let's think of this. Class is done today, and it's a nice day, so I need to go shopping. I'm going to go run to Costco or something like that. And on my way to the car, as I'm getting in the car, I slam the car door on my hand. By the time I get to Costco, what's happened to my hand?
Swollen.
It's swollen up like a balloon. I have edema, swelling, an accumulation of fluid in that space. Yeah, it makes it a little harder for me to flex my fingers. It may be a little painful, right? It's going to be a little harder for me to get my ring off when I get to the bar and those kind of problems, but it's not a major issue. But what happens if you get swelling in here?
Messes everything up.
That is really, really bad. So we want to make it as easy as possible to get that blood out of the cranial cavity. So by having these big, huge, loose veins, it makes it very efficient at draining the blood, draining the fluid out of our cranial cavity. And this is going to play a major role in helping to reduce swelling. Reduce that edema to make sure that it doesn't happen. All right. Excellent. Questions on the dura? All right. Notice under the dura is a second layer, and that second layer is known as the arachnoid matter. Arachnoid refers to what? If you have arachnophobia, what are you scared of?
Spiders.
Right. Notice this layer has a lot of, oh, the purple doesn't show up at all. A lot of tiny little fibers that have kind of a cobwebby appearance that forms this space. And this space underneath the arachnoid matter is what is known as the subarachnoid space. And what is that subarachnoid space filled with?
Cerebral spinal fluid.
Filled with cerebral spinal fluid. This, as we talked about, provides cushion and protection but it also can provide nutrients and removal of waste of the nervous tissue. This is really, really important. Notice the dura mater sits right on top of the arachnoid matter. As we've talked about before, when I put my two hands together, do they become just one structure?
No.
No. So, but there really isn't anything between them. Does anybody remember what we called that?
Potential space.
Yeah, potential space. Notice here, we have a potential space between my hands. Notice here, we have a potential space between the dura mater and the arachnoid matter. There is nothing in a healthy brain between the dura mater and the arachnoid matter. This is a potential space. And that potential space is called the subdural space. Now notice I said in a healthy individual, there is nothing between there. However, if, right, because I have forgotten to take out the garbage for yet another time, and I take a blow to the head from a hammer from some random person in the house who won't be named, right, that damage could cause blood vessels to rupture, and I could get a swelling of blood that would occur within this space. And suddenly that potential space starts to fill up with blood. And what would we call that condition? Come on, I know you know. What do you call a swelling of blood in the subdural space? You say again, Emily?
Subdural hematoma.
Exactly, a subdural hematoma. Exactly. So notice with some type of brain injury or cranial injury, cranial trauma, let's say it that way, you could get a subdural hematoma. You could get fluid filling that space, but normally in a healthy individual, there's nothing in that space. It is just a potential space. The subarachnoid space is an actual space filled with actual stuff. All right, subarachnoid space contains cerebrospinal fluid. And lastly, notice there is a third layer to our meninges. The brain has this wonderful shrink wrap on its outer surface. Notice this goes down into all of the sulci, stabilizing the gyri, holding them in place like shrink wrap. Remember, as we mentioned, nervous tissue, when it hasn't been fixed, has that soft, flexible consistency of like a Jell-O jiggler. So we need to provide some integrity, provide some structure to it. And so putting this shrink wrap around it helps to stabilize it and hold it together. It helps to stabilize and hold blood vessels in place and provides a little protection and a little structure. And this is known as the pia matter. Notice, I'm going to cheat. I closed it, but I'm going to go back to it. There you go. If you remember, we looked at this picture when we were first talking about the lobes of the cerebrum. Here, we have a brain that has been removed, and the dura mater has been removed as well. But notice, we can still see all these big blood vessels, superficial blood vessels in place. And notice how shiny the surface of this nervous tissue is and how compactly held together the gyri are. This brain still has the pia mater on it. That pia mater is a shrink wrap sitting right on top. But like a shrink wrap, it doesn't connect to the tissue. So if you're really, really careful, you can remove the pia mater. When you remove the pia mater, notice all the superficial blood vessels are gone. There's still some deep blood vessels. But notice now the nervous tissue has not been damaged, but we can now see how it's starting to open up a little bit. All those gyri are forming more open spaces because we don't have that shrink wrap on it anymore. We can see this a little bit. Come on. There we go. In this picture here. Notice here on the right, we can still see part of the intact hair, scalp, bones of the skull. And then here we see that big, huge, thick, fibery, leathery dura mater. Notice in that dura, we can see some of the spaces where they've been opened up to form those sinuses. And notice over here, the dura mater has been removed. And when the dura mater has been removed, notice we can see that nice shiny surface and all the gyri being held together by the pia mater. Notice what we're not seeing is the arachnoid matter. The arachnoid matter is much more flimsy. Notice if you kind of look carefully down here by the spinal cord, you can actually see how it's a little fuzzy in this area here. That little bit of fuzziness in that area there is a few little bits of that arachnoid matter that's around the spinal cord and around the medulla oblongata. But it's not very, it's a very flimsy tissue. So typically it's the dura mater and the pia mater that are more obvious. The arachnoid matter is really more about forming a space that we can put that cerebral spinal fluid in. All right. Questions on that? Notice two things I want to strongly emphasize. Clearly, there is no space between the dura mater and the bone because the dura mater is part of the periosteum and they're connected to each other. And remember, there is only a potential space between the dura and the arachnoid matter. They're not fused together into one thing, but there's also nothing in that space. It is a potential space. I'm making a point of emphasizing this because when we talk about the protections of the spinal cord, we'll see the spinal cord also has a dura mater around it, also has an arachnoid matter around it, also has a pia mater around it. But there is a huge difference between the cranial cavity and the vertebral cavity. What is the big difference about the bones that protect the brain versus the bones that protect our spinal cord?
The spinal column is open. It's not closed off completely.
True, it isn't closed off completely, although you can argue there were plenty of holes in the skull too, right?
It flexes.
There you go, movement. The bones of the skull do not move. The bones of the vertebral column do. So while we're going to have these three layers of meninge, there's going to be some slight arrangement changes to allow for that flexibility of movement so that we can move our vertebral column without damaging the spinal cord. We don't have to worry about movement up here because the bones of our skull don't move. We do have to worry about it in our spinal cord. Excellent. All right. Lastly, one great picture. I love this picture from your textbook that does a great job of showing all of these layers we've been talking about. Notice layer 1, layer 2 of the dura mater. This happens to be one of those areas where there's space in between them. And when there's space in between them, that is filled with blood, forming that dural sinus. Notice here we have that subdural space, which again, as we know, is a potential space in the skull. Our arachnoid matter produces our subarachnoid space, which is filled with cerebral spinal fluid. And then here is our pia mater, that shrink wrap right on the surface of the cerebral cortex. We did that. Remember, I mentioned not only does the separation of the dura mater give us our fluid-filled spaces, those sinuses that we see here and over here to the side, and this one here and this one down here and so forth. But remember, we also talked about how they fold in. And by folding inward, they help to stabilize and hold the brain and the brain stem in place. And because anatomists hate us, we know that any of these folds are going to have names. So notice here, as we mentioned, we have that fold located in the longitudinal fissure between the hemispheres of the cerebrum. So let's actually put that there too. We call that fold of the dura in between the hemispheres of the cerebrum in that longitudinal fissure, the falx cerebri. And make a point of identifying it that way because notice also there is a fold between the hemispheres of the cerebellum. Notice it's not anywhere near as big because this doesn't go between them. It sits on top of the vermis. Remember, the vermis sticks out. It doesn't fold in. So it's a much, much smaller fold, but this smaller fold is still going to get a name. And this smaller fold is known as the falx cerebelli. So falx cerebelli is between the hemispheres of the cerebellum, and the falx cerebri is between the hemispheres of the cerebrum. Notice there is one more fold in the dura mater. This one basically, its job is to separate or really stabilize the space between the cerebrum and the cerebellum. Notice this sits like a tent over the top of the cerebellum. Which is where it gets its name. Its name is the tentorium cerebelli. So 3 big folds of the dura mater that help to stabilize the cerebrum and the cerebellum in space so that every time you move your head, your brain isn't rattling around inside of your skull. All right. Questions on that? Excellent. Oops. There we go. We did that. Perfect. The last protection we have to talk about in relationship to the brain and spinal cord, and vertebral column, not vertebral column, sorry, and brainstem, brain and brainstem, is the spaces in between our ventricles. Notice this great picture from your textbook does a really nice job of showing these. Notice we have the 2 C-shaped side-by-side lateral ventricles on the 2 sides. Then right in the middle between the 2 thalami, we have our 3rd ventricle. And notice there's actually a void in the middle of the 3rd ventricle. Why is there a small part of the 3rd ventricle that doesn't have fluid in it? Why is there an island in the middle of my 3rd ventricle?
For the structure that's connecting the 2 thalami.
Yeah, exactly. And what was that bundle of axons that connects the 2 thalami together?
Intermediate mass.
Intermediate mass. Excellent. From there, notice we travel down that cerebral aqueduct or aqueduct of the midbrain into that fourth ventricle that was located between the pons and the cerebellum. So we've talked about all of these structures and most of these structures. Here we see what they would look like inside of the brain from a side view. Notice we can do the same thing from a frontal view. And again, now we clearly very nicely see the two lateral ventricles. We know that septum pellucidum is sitting right here in between them, separating them, keeping them apart from each other. Then here is our third ventricle between the two thalami, cerebral aqueduct, into our fourth ventricle between the pons and the cerebellum. Although it's really cool is if you were to take a person's brain, drill a hole into the top and fill it with plastic, let it harden, and then you peeled away all of that pesky nervous tissue, what you would get is a model that looked like this. Do they have both views? Nope, just the one view. So basically this model is a model of the spaces, like using those spaces as a mold. Notice we have our C-shaped lateral ventricle here. We have our third ventricle here, cerebral aqueduct into our fourth ventricle here. I've put some pink all over this, but if you notice closely, there is some pink located here in the fourth ventricle, some pink here at the top of the third ventricle, and a little bit of pink up here in our lateral ventricles. Did whoever was using this leave a little bit of chewing gum on them before they took the picture?
No.
No, that pink stuff represents a substance known as choroid plexus. Choroid plexus is basically a specialized capillary and something else. And it is responsible for making our cerebrospinal fluid. When we were learning about the support cells, didn't we learn about a neuroglial cell that played an important role in the making of the cerebrospinal fluid?
Ependymal cells.
Excellent. And that's what choroid plexus is. Choroid plexus are these specialized capillaries lined with ependymal cells that are responsible for making our cerebrospinal fluid. And it is found in all four ventricles. All four ventricles have this choroid plexus in it. So cerebrospinal fluid can be made in any of our four ventricles. Notice if we go back to our lecture, notice here, this illustration cheats, peeling away the septum pellucidum so we see into the lateral ventricle. And sure enough, inside here in our lateral ventricle, we see some choroid plexus. Here in our third ventricle, we see some choroid plexus. And here in our fourth ventricle, we see some choroid plexus. So choroid plexus can be found in any of our, will be found in all of our four ventricles. So cerebrospinal fluid can be made in any of these four locations. So if you are a drop of cerebrospinal fluid that is made, are you only made in one location?
No.
No, you can be made in any of these. But for argument's sake, and to help us understand all these spaces, Remember, all these things we are identifying right now are spaces that are filled. And since I've written it twice now with cerebral spinal fluid, I'll just abbreviate it like that. These spaces. This isn't the path that every drop of cerebral spinal fluid is going to make, but it is one of the potential paths it could make. Let's say, for instance, you are a drop of cerebral spinal fluid that is made in a lateral ventricle. From that lateral ventricle, there is only one place you can go. You have to get into the third ventricle. Notice if we go back 2 pictures, here is our lateral ventricle, and we need to get into the third. And luckily for us, there is a small opening that will allow us to do that. But I think you see this actually best from this view. So again, you're this drop of cerebral spinal fluid made here in the one of the lateral ventricles. Notice they're both lateral ventricles, left and right. One is not 1 and one is not 2. They're just the lateral ventricles. You don't want to fill this space up till it bursts like a balloon, so you need to be able to leave. So you need a space that travels from one ventricle to the next. And lo and behold, that space that allows you to get from the lateral ventricle to the third ventricle is the interventricular foramen, which sounds like a lot of vocabulary, but interventricular means between the ventricles. Foramen we know is a space, so its name tells you exactly what it is. It is a space between the ventricles. Once you're in your third ventricle, we can swirl around inside of here. And remember, there is going to be that void, whoops, wrong direction, that void in the center of it where the intermediate mass is. So I can produce some turbulence as I'm roaming around in there. But remember, as we mentioned, there is only one place to go from here into the fourth ventricle. This diamond-shaped ventricle between the pons and the cerebellum. And there's only one way to get there, the aqueduct of the midbrain. To this point, I think it's pretty simple and straightforward, but I still want to go to the whiteboard so we can move forward from this. So, and again, I'm going to do this super simple. These are our lateral ventricles. I'm probably a little too big. From the lateral ventricles, there's one way and only one way to go. And that is into our third ventricle. And the way you get from the lateral ventricle to the third ventricle is the interventricular foramen. So if you're if you're a drop of cerebral spinal fluid and you're made here in a lateral ventricle, the only thing you could do is go through the interventricular foramen into the third ventricle. Now you're in the third ventricle because you came here from the lateral, or you were made in the third ventricle. It doesn't matter. There's still only one place you can go, into the fourth ventricle. That diamond shape ventricle, and there's only one way to get there. Cerebral aqueduct, or what is also known as the aqueduct of the midbrain. So you're a drop of cerebral spinal fluid here inside of the fourth ventricle, because you were either made through the third or made in the lateral, or you were made here in the fourth. We don't really care how you got here. We just know you're here. And notice there's been only one route you could take up to this period of time. But here in the fourth ventricle is where we have choices. There is only one way in. And what was that one way in?
Cerebral aqueduct.
But there are 4 ways out. The first 2 of these ways are off to the side. And actually, I'm going to draw these in red because it amuses me to do so. There are 2 openings to the sides. Not surprisingly, these openings to the sides are known as the lateral apertures. They exit the fourth ventricle and basically lead into the subarachnoid space. And they do that above the cerebrum. Or cerebellum, sorry. And of course, there's 2 of them. So 2 of the ways out are the lateral apertures. The third option is actually to continue out of the brainstem and actually travel down into the center of the spinal cord. This is a space known as the central canal. Now, obviously, the goal isn't to fill up the spinal cord with cerebral spinal fluid. So when you get to the bottom, it actually leaves and ends up into the subarachnoid space. So, central canal. And the third way out, and it's going to be tricky for me to draw it. Nope, don't want it to be green. The third way out is right at the bottom of the fourth ventricle, right on the midline, which is why it is called the median aperture, is an opening that exits below the cerebellum on the midline and goes into the subarachnoid space. And so that is our fourth way out. Median aperture. Notice now that we've done this in this very simplistic diagram, let's look at the pretty pictures. Notice here when we look at the spaces from the front, we can see the 2 openings here. Notice above the cerebellum to the side. Those are the lateral apertures. Notice they've put a little hole right here to represent that back opening on the midline below the cerebellum, the median aperture, and we can see that passageway down into the spinal cord, our central canal. So this picture shows all of those openings, the 4 exits, and of course the 1 entrance to our fourth ventricle. Notice if we go here, they show those as well. Notice we can see the central canal traveling through the spinal cord, and notice coming out the bottom into the subarachnoid space. Here's our median aperture to the back below the cerebellum into the subarachnoid space. And they put an arrow here where a little dot would be going out to the side that would come into the space above the cerebellum. Notice one more nice picture. Oh, wait, no, that's not the one I wanted. Where's the one I wanted? This is the one I wanted. Notice here from our posterior view, we very nicely see our fourth ventricle. Notice we see the choroid plexus in our fourth ventricle. The two lateral apertures would be to the side. The central canal travels down the center. And then coming out here to the back would be that median aperture. So we see that really nicely. There are options with the fourth ventricle. Here we see that up close. Notice again, there's that lateral aperture to the side coming out above the cerebellum, median aperture below the cerebellum, down into the spinal cord, lateral ventricle, interventricular foramen, third ventricle, cerebral aqueduct, fourth ventricle. All of them have cerebral choroid plexus in them for making cerebral spinal fluid. And this does a nice job of showing. And then also remember we looked here. Here we saw the third ventricle and the intermediate mass and the two lateral ventricles. Clearly we're not going to see down into the fourth because this is a superior view. All right. So those are all of the spaces associated with the cerebral spinal fluid. And notice one last thing. Regardless of how you leave the fourth ventricle, whether you go out a lateral aperture, whether you go out the median aperture, or whether you go all the way down the spinal cord first, everything, all of the cerebral spinal fluid ends up in the subarachnoid space. Which is awesome because that is going to surround the brain and be able to bathe it and protect it. But it also means we need to be able to get rid of it. And notice we peeked at it quickly, but I didn't emphasize it, but we see it here. There are finger-like extensions of the arachnoid matter that stick out into those dural sinuses. As we learned in the skin, a finger-like extension is called a villi, or the singular being villus. So we have these arachnoid villi, or what are also known as arachnoid granulations, that stick out into that dura, allowing us to release that cerebral spinal fluid back into the blood that it came out of. And in fact, I jumped over it, but notice here, we see that same pretty picture we saw before. Dura matter with a dural sinus in between, arachnoid matter, pia matter, subdural space, potential space. But notice here, some of that arachnoid matter sticks out in a finger-like extension, arachnoid villi or arachnoid granulations, that releases the cerebral spinal fluid back into the blood where it came out of. And that is how we protect the brain, and that is how we produce and circulate our cerebrospinal fluid. All right. Questions on that? All right. I have no idea what we are going to do next. I need to look at our lecture because we have basically two choices at this point. we either now need to start talking about our spinal cord, or we need to talk about our cranial nerves. I think I want to do the cranial nerves first, but I need to look at the two lectures to see what I think is going to be best to cover first. So while I do that, let's go ahead and take our second break, and I will figure that out. Grab your cranial nerve handout if you haven't already, just on the off chance, because I think this is where we're going to go next. I just need to double check to make sure we don't have to do the spinal cord first. But I think it's more beneficial to do the cranial nerves. I just need to make sure that there isn't something in the spinal cord we need to talk about before we do the cranial nerves. So I have to figure that out. So let's go ahead and take our second break. We will restart at 10:45. Start the recording at that time. And like I said, grab your cranial nerve handout if you haven't already, just on the off chance, because I think that's what we're going to do next. I just need to double check that. All right. Any questions before we take our second break? All right. I will see you in 15 minutes. All righty. We have made it through the first half of our cranial nerves, and I apologize for those of you who missed some of that because we did not record it. Again, like I said, please feel free to look back at previous lectures, and again, your book does an excellent job of going through these things as well, so you can do that as well. So we made it to the top of the mountain. We're working our way back down and on to our facial nerve, cranial nerve 7. What is the functional type of cranial nerve 7?
Well,
Yeah, it's a mixed nerve. It's both, absolutely. Or again, let's be consistent, both. So that means it is going to have both motor and sensory functions. Motor is where it gets its name. Its motor function is basically to control the facial nerves, the facial muscles. Again, as we talked about, we are very good at nonverbal communication, especially with our facial expressions. And that ability to look angry, sad, depressed, aroused, whatever, without saying a word is due entirely or primarily to the function of this facial nerve. If you remember, we talked about this facial nerve once before, and its function in controlling the facial muscles, and that was in relation to its skull exit. Notice its skull exit is this tiny little space, this tiny little hole between the big round bump on the back of the head and the big sharp bump. What was the big round bump of the temporal bone called again?
mastoid process.
And the tiny pointy one?
Styloid process.
Excellent. So the hole in between it, its skull exit, is what?
Stylomastoid. Foramen.
Yeah, it is a foramen between the styloid process and the mastoid process. It is the stylomastoid foramen. If you're in the classroom, one of the things you would have done is taken one of our wooden probes that you use to, explore the holes of our skull with, and you would have found that it doesn't fit in this hole. The stylomastoid foramen is very, very small. And as we talked about, the problem with that is that we have this big solid feeling mastoid process that, as we know, actually has a bunch of air-filled spaces inside called the mastoid sinuses. And as we talked about, if there is inflammation to those mastoid sinuses, that inflammation can actually squeeze and narrow that opening. And as it squeezes and narrows that opening, it can squeeze the nerve so that it becomes non-functional. And suddenly now basically everything from your eyebrows down, you can no longer move. So you go to smile and only half of your face goes up and the other's just kind of droopy. What did we call that condition again? Anyone remember? The name doesn't ring a bell by any chance? Seriously, that didn't get it to you?
Bell's palsy?
There you go, exactly. Bell's palsy. Exactly, that Bell's palsy is that condition where you get that, that basically inactivation of the nerve, commonly by swelling in the mastoid process. And if you remember, I told you there have been at least 3 documented cases where this occurred because someone was driving in the car with the window open. So the air was flying past the air, irritated the mastoid sinus, which became inflamed, squeezed the nerve, paralyzed the face. The good news is it can typically be resolved pretty quickly, especially with some anti-inflammatories and things along those lines. But in some rare instances, it can have a prolonged effect where it can last for months. And if the damage to the nerve is severe enough, the palsy can actually become permanent. Now that is its, I wonder if I have the next slide. This works. That is its primary motor function, or at least its somatic motor function, controlling that facial expressions. There is also some autonomic motor. For that autonomic motor, it plays an important role in mucus production in the nasal cavity and our production of tears. So it affects those glands, which is why they're autonomic, because that's not something we voluntarily control. Right? Most of us can't cry on command or make our nose run on command. But there is also a sensory function. And that sensory function, as we see here, is basically taste from the anterior 2/3 of the tongue. Nope, I thought I had it, but I don't. Okay, excellent. I don't have a picture of the tongue, so let's draw a picture of the tongue. Right, again, knowing what an amazing artiste I am. Again, you're going to, of course, be shocked by how realistic this tongue looks. There you go. There is our tongue. If you were to look at your tongue, or yeah, let's actually look at one of the charts. I bet they'll have it here. So if we go back, nope, not what I wanted. I want that. My guess would be digestive is probably the easiest place to see it. That's not bad, but this is the picture I want. Notice here we have the tongue. Notice about 2/3 of the way back from the anterior surface of the tongue are these big, huge, large papillae. These big, huge, large papillae are what are known as the circumvallate papillae. Notice behind that, the tongue is primarily covered with our lingual tonsils. Those lingual tonsils are, along with our palatine tonsils, providing that defense from any harmful material that may be coming in. And when you stick your tongue out, it is primarily the anterior 2/3 of the tongue that you stick out. There are lots of little bumps all over the surface of the tongue, and these big bumps, called the fungiform papillae, because after all, they are finger-like extensions that stick up. And those circumvallate papillae are the ones where we perceive our sense of taste. And how many taste sensations do we have?
Either 4 or 5.
Which one? You are correct. It is either 4 or 5.
I think it's 4.
How many?
5.
There you go. Excellent. We have 5 taste sensations. Excellent. What are they?
Sweet,
Sweet,
salty, sour,
Bitter. Umami.
There you go. Again, notice your guys' hesitation on 4 versus 5, because originally it was 4, and then more recently, some Japanese researchers found a 5th taste sensation that provided like kind of that sensory taste sensation, like from the juices of red meats or MSG, provides a very sensory sensation. And of course, the Japanese word for sensory, for savory sensation is umami. So we have those 5 taste sensations. And anyone who has ever been in a kindergarten class where they have those pictures of the wall of the eyeball and of the skin and of all the other things, you've seen that on this, they have this very precise map where, for instance, the sweet sensation is located up here at the tip, and salty is along here on the sides, and sour is in the back. And they have this very elaborate map of how these taste sensations are represented on the tongue. However, the kindergarten classroom isn't the only place that they talk about these types of maps. The other place they talk about these types of maps are in Williams-Sonoma. Because if you go hang out at Williams-Sonoma for any length of time, they'll come up and start talking to you, and you can speak to them about their Riedel wine glasses. Riedel wine glasses are these very, very beautiful wine glasses that cost about $30 a wine glass. Now, how can a wine glass cost $30? It's simple. There's 2 key specializations to them. The one is that each wine glass is made specifically for the type of wine that you're going to drink. So it has a different shaped bulb to it so that when you swirl your wine, the right amount of air is added to it to maximize the taste sensation. But what really makes these wine glasses worth $30, $40, or even $50 a piece is that their lips are set in such a way so that when you take a sip of the wine, it presents the wine to these taste sensation regions in just the right order to maximize your enjoyment of the wine, making that amazingly expensive wine taste even more amazing. And of course, for doing something like that, it's worth every single penny of those $50, right?
or you can just put the wine glass right into the bottle and just drink it straight from there.
True, you could do that. And also, there's also the fact that it's all complete BS. Do you have a taste map on your tongue? Absolutely. After next week, when you're done with all your exams, a very fun activity that you and your loved ones can do is sit around a table, right, with a bunch of paper towels and a bunch of little bowls of solution. Take a Q-tip, dip it into some sugar water. Take a paper towel, dry off your tongue, and then take that Q-tip with the sugar water and wipe it along the surface of your tongue. And when you do that, you'll notice that you're only able to taste sweet in certain regions of your tongue. Rinse your mouth out, dry it with a paper towel, and then do the same thing with salty. Some salty water. And you'll notice that there are parts of your tongue where you can only taste the salty. And then lemon juice for sour, coffee for your bitter, MSG if you're not intolerant to it, or like I said, the juices of some red meat if you're not vegetarian. Those are things that can be used for that umami sensation. And what you'll find is you absolutely do have a taste map on your tongue, but everybody in your family has a different one. No, not everybody's sweet is in the front, not everybody's bitter is in the back or anything like that. We all have our own unique, distinct taste maps. So those wine glasses like Riedel at Williams-Sonoma are complete BS. Right, yeah, just use your red Solo cup. All right, but The point is that sensory sensation comes in from our 7th cranial nerve. So our 7th cranial nerve is what carries all of this taste sensation into our brain. Now, notice one last thing. The one thing we didn't mention here is spicy. Why isn't spicy a taste sensation?
Is it nasal? Does it have to do with the?
Not a bad guess. It isn't so much that it does with nasal, although you are absolutely correct. What we are able to inhale from the moisture. Okay, let's do this. Okay, our chemical reception, our perception of chemicals in our body are actually those chemicals when they are dissolved in water. Okay? So, for instance, when you smell something, those molecules are in the moisture of the air, and so we're able to perceive them when they do that. The same thing with our taste sensation. Your ability to taste things is because you're dissolving those chemicals into your saliva. But doesn't your saliva have a lot of stuff in it as well? Yes. And it turns out your taste buds are actually specialized for your saliva. There are gustatory neuroanatomists, a very, very odd group of individuals, but they've actually studied this. What they will do is they will take some of your saliva and they will dissolve a chemical in it. And then they will take someone else's saliva and dissolve a little bit of chemical in it. Then, using that Q-tip method we talked about, they will take some of your saliva and spread it over the surface of your tongue, clean it off, and then take some of somebody else's saliva and spread it out over the surface of the tongue. And in doing this, they found that people are much better at detecting chemicals when they are dissolved in their own saliva than from somebody else's, which is why I tell my daughter not to swap spit with other people. All right. But as you also mentioned, if your nose is plugged and you can't get that air in there, then it dramatically does decrease your sense of taste. So sense of taste and sense of smell, both of which are chemoreceptors, are related to each other and they do enhance each other. And what's really amazing about this is how that chemical gets into your nose makes a difference. For those of you who have ever gone to France or ever eaten really funky cheese before, when you smell that funky cheese, it is really, really repulsive. But when you eat it, does it taste as bad as it smells?
No.
No. And the reason for that is once it's in the mouth, the air, the chemicals mixed with the moisture actually come up the back of the throat to the nasal cavity that way. And when those same chemicals come up from the saliva, as opposed from coming from the outside world, they smell differently and they affect the taste differently. And so it smells different when it's in your mouth than when it's outside of your mouth. And tell you, the brain is a cool thing. All right, excellent. But none of that has anything to do with spicy. Where does spicy come from? Well, remember, these are chemoreceptors. We have a chemical that binds to a receptor. This is certainly something we understand about taste because our beverage companies spend gajillions of dollars to find just that right chemical that can turn the key of those sweet receptors you have so your diet soda tastes like there's sugar in it when there's truly no sugar in it. Right? That's what artificial sweeteners do. They try to trick this sweet by turning that key to make you perceive something sweet. Well, spicy is the janitor's key of chemicals. And what is a janitor's key open?
Everything.
Everything. And that's what happens. Spicy stimulates all these taste receptor types at the same time, causing a big, huge neural signal. And that big, huge neural signal is confusing and is perceived as pain. Plus, it also stimulates the tactile receptors, like that trigeminal nerve, stimulating them, making you think that you are being damaged, stimulating a pain response. That's why your face gets red. That's why you sweat, because that spicy is tricking these tactile sensations into thinking you are being burned. It's stimulating a pain response. And so you get inflammation, you get that redness, you get that sweating as a response to that. So basically, this janitor key is unlocking a whole bunch of doors, causing a whole bunch of problems, which is where we get that sense of spiciness. All right, questions on our facial nerve. And of course, remember, as we mentioned, its skull exit is that small little hole between the styloid process and the mastoid process, the stylomastoid foramen. Now, there is one tiny issue. If we look at the inside of the skull, do we see the stylomastoid foramen?
No.
No. But if you do remember, there is an opening right here on the side of the temporal bone known as the internal auditory meatus or canal. Not surprisingly, that is where cranial nerve 8 is going to go. But remember, 7 and 8 are BFFs. And in fact, if we switch to our next picture, lo and behold, sad, lonely 6, 7 and 8 are our BFFs. And not only BFFs in that they hang right next to each other, but they both enter the skull through the internal auditory or internal acoustic meatus. Now 7, notice they've cut, is then going to go and eventually leaves. And so it exits the skull in the stylomastoid foramen. But it actually enters the skull through the internal acoustic meatus. Now, is where it enters the skull one of the questions you are responsible for on the exam?
No.
No. So all we care about is where it exits the skull. So for 7, our skull exit is the stylomastoid foramen. I make a point of emphasizing this because we are now on to cranial nerve 8. Back in ancient times, and by ancient times, I mean when I was in graduate school, 8 was known as the auditory nerve. And in fact, my not suitable for work mnemonic, which no, I will not share with you, for cranial nerves uses an A in the 8th spot because it used to be the auditory nerve. However, the auditory nerve is a horrible, horrible name for this nerve because this nerve does 2 equally important things. There are actually 2 branches that provide 2 different types of sensory information. Yes, one of those types is perception of sound, hearing, but also the second one is our equilibrium and our balance. If you are in an elevator that has no windows, you step into an elevator, you don't push a button. You just sit in there and wait for it to start moving. And it has no windows. When it starts moving, could you tell whether it's going up or down?
Yeah.
Yeah. right? And even if it went left or right, would you be able to know that as well?
Yes.
It'd be really weird for an elevator to do that, but you would know it was doing it, even if you couldn't see out. That's what we mean by equilibrium. And if I put you here in my office chair, which conveniently enough spins, and I spun you around 16 times, and then I told you to run downstairs and get a drink of water, would that be something that would be easy for you to be able to do?
No.
No, because I would be upsetting your balance by doing that. So there are two equally important types of sensory information that gets to our brain through cranial nerve 8. And there are two special sensory structures that allow us to do that. The sensory structure that allows us to hear is known as the cochlea. And the special sensory structure that gives us our equilibrium and balance includes a big structure known as the vestibule. Here in the picture, we see these structures. This big coiled snail shell looking structure is the cochlea. This big block-like structure over here is the vestibule. So rather than the disservice of going with this name that really wasn't indicative of what it does, instead, they changed the name of this to emphasize the two special sensory structures. So cranial nerve 8 now is much harder to spell, vestibulocochlear, but like most of the alphabet soup names that we are responsible for, its name tells us what it is. It receives information from the vestibule and from the cochlea, giving us our hearing and our equilibrium and balance. Now, not surprisingly, that means its functional type is what?
Sensory.
Excellent, it is indeed sensory. And the only trick is technically, does cranial nerve 8 leave the skull?
Yes.
Well, technically it doesn't leave the skull because it goes into the temporal bone, but it does leave the cranial cavity. So you're right. Yes, it leaves the cranial cavity. But notice, unlike every other cranial nerve, it's not going out the bone to go somewhere else. It stays inside the bone. But you are right. It does leave the cranial cavity. And so we use that exit from the cranial cavity as its skull exit. And of course, as we already talked about, that is the internal auditory meatus. All right. Questions on that? All right, we have run out of time, but again, that was one of the reasons why I wanted to emphasize doing this today. So if we didn't finish it today, we could finish it tomorrow, and you would have all the information going into this weekend. So we still have 9, 10, 11, and 12 to do. We will do that at the beginning of class tomorrow, and then from there we will go on into our spinal cord, finish off the central nervous system, and we should have enough time to do all of that, or most of that, tomorrow. All right. Questions on any of that? You know how to play this game. There's no reason you can't go and take a look at least 9, 10, 11, 12 on your own. See if you can fill out the rest of your chart, but we will go through it together to make sure that you filled it out correctly. All right, any questions on any of that? Again, I apologize for the error of not restarting the recording. Again, I would ask you, if you notice that I haven't restarted it, to please point it out so that we can record that. But I guess this is the advantage of being here. You get the information, and anybody who's not here missed out on it. But again, that's not why we did it. That's not the importance, because again, I know we cover a lot of material, and I want you to be able to go back and revisit this material and hopefully it makes sense. But at least you have previous versions of this lecture, and at least you have your textbook, which does an excellent job of doing that. So I apologize for that error. Help me catch it if it happens again. All right, questions on any of that? All righty, then excellent. Have a good day, and I will see you bright and early tomorrow morning.
Thank you.
Thank you.